Thank you, Dr. Samira, for this presentation. My question about the correlation between microbiology results, like the culture and other results, with some blood tests like procalcitonin and CRP and some cytokines. Do you have that in your lab to decrease the time of detection and diagnosis of sepsis?
Yes, these assays are available at the chemistry lab, and at one point we did the correlation. We don't do it so routinely. Yes, it has an value in telling the patient is septic or not. But again, sometimes if the patient is so sick or deteriorating so rapidly despite on antibiotics, sometimes they need the results, what's going on, mainly this antibiotic susceptibility that we are facing. We have no problem in identifying the organism nowadays in a very short time. It's just the antibiotic susceptibility.
So is there a single test you recommend for diagnosis of sepsis? Because we are now working with
I think yes.
With my group now, we are working trying to find out some markers and blood markers for early diagnosis of sepsis, which can be done by the nurse and the bedside of the patient. Doesn't need to go to each time to the lab and waste the time for lab, because I think and everybody know that every delay of 1 hour of sepsis, it will increase the mortality 15%.
That's true. It is a point of care assays, yes, and I guess if there is an earlier results for the clinician, it always be valuable. But you remember, you have to add in your system your antibiogram, so people would know what is the type of organism and antibiotic susceptibility pattern to use.
Thank you, Dr. Samira. We're going to have a break, 15 minutes for the coffee break. But I mean, before the coffee break, we would like to give some certificate to the speakers, Dr. Yazid, Dr. Khalid. The first one is Dr. Walid. Dr. Samira. With the speaker, I think he left for the coffee break. Dr. Abdul Wahab, we're waiting for him. Our coffee break, you have to go downstairs. Dr. Abdul Wahab. Our coffee break will go downstairs, and they will give you assistance. You have to go downstairs. Under the stair, there is an elevator. Before the elevator, on your right-hand side, there is, I think, recreation for the student recreation. Just go through this double door, and you will find the coffee break there. I'll give some of the... Now we're going to start with our next session in the lecture for today, and we're going to start with Dr. Yazid al-Sheikh. Dr. Yazid al-Sheikh is Associated Professor in Medical Molecular Genetics, graduated from SSCLS KSU, for a bachelor's degree, master in human molecular genetics, and PhD in cancer genetics from Glasgow University, former head of clinical lab scientist, KSU, and former president of the Saudi Society for Clinical Laboratory Science, and now the dean of the College of Applied Medical Science at King Saud University. Doctor.
Shukran, Doctor. Bismillah ar-Rahman ar-Rahim. My talk today is going to be on the subject of CRISPR and whether it is a new thing or a new tool in diagnostics. First of all, I'd like to have a sense of the audience. Have you ever heard of CRISPR before? Can I see some hands? Afa. أنا كان ودي أخذ راحتي في الكلام بس يبدو لي الآن المسألة محكورة شوي. Anyway, this is the tool. So what I'll try to do is introduce the CRISPR system and then go on and introduce the CRISPR Cas9 adaptable programmable system. And it is actually the actual editable programmable tool in the CRISPR cascade, or one of them. And to discuss at the end whether this is a viable tool in diagnostics. So what is CRISPR? Most scientists who are familiar with the technology associate CRISPR with designer babies. And yes, it is possible. It is possible to actually manipulate the genome of embryos using this technology. And even at its infancy, we are at the very start of the CRISPR technology, or the CRISPR era, and people are embarking on trying to manipulate human genomes. Now, we hope that the intent of this is for medical purposes, for curative purposes, defeating disease, having children that are disease-free, try to conquer and manipulate our biology. And what we mean by conquer is actually having the tool to actually make a difference in our biology. Manipulating our genome exists. The techniques are there. They've been there for a long time. But they're a very crude system. You cannot reach the targets that you want efficiently. You cannot do what you want efficiently. But CRISPR changes that fundamentally. It could be used as a tool for increasing life expectancy, life quality, and it could be abused. You could use this tool to, such as we have here, to create a breed of super soldiers, for example, or people with preferred traits that are not necessarily ethical. So the story begins with the oldest war in the world. The oldest war on Earth is the war between bacteria and virus. And each day, today, till this day, viruses eradicate 40% of the world's bacteria. And as a result of this war, the bacteria, bacterium and archaea developed a sort of an immune system to combat this viral infection, particularly phage infection, bacteriophage infection. So the bacteriophage, which is the arch enemy of bacteria, inject their DNA into the host bacteria and replicate within the host cell. So this system was developed by bacteria as an adaptable or an acquired immune system. So CRISPR is short for clustered, regularly interdispersed, short palindromic repeats. It's a mouthful, but once you get used to it, it's not so bad. So early in 2005, through sequencing of E. coli genome, scientists discovered that the bacterial genome had these two regions, a set of Cas genes and a set of cluster of repeats interdispersed with different spacers. And these spacers were later identified to be viral in origin. So these Cas genes also encode for a set of different genes, but their function was not known. So it was only recently in the Dowdena lab, around about 2012, that the function of these genes were found. And it basically says that once a bacteriophage injects its DNA within the host cell, this system, the Cas genes, which is CRISPR-associated protein genes, and the CRISPR locus itself, act together to effectively combat each bacteriophage infection independently depending on its sequence. So with time, with each infection, the bacteria formed a sort of a library of all past infections. So the Cas or the CRISPR-associated genes encode for different types of enzymes, particularly helicase and endonucleases, and form some sort of a hybrid enzyme that has both properties, the properties of endonucleases and helicases to unwind DNA and endonucleases to cut DNA. And downstream to that is the repeat system or the repeat cluster interspersed with different spacers. Now what happens is that when the phage injects its DNA, and this is a known infection, a previous infection that is found in the library, the Cas genes will transcribe a protein accompanied with the corresponding sequence of the past similar infection to form this CRISPR-Cas molecule and readily combat it. However, when you have a new system, a new sequence of DNA, a really new virus, what happens is that the Cas genes produce a class 1 Cas gene, a Cas protein, which will actually act like an antigen presenting cell. It will break down the DNA and present it to this cluster repeat and include it to the library. So this is the class 1 CRISPR protein. It will degrade this new DNA and include it in this ever-growing library. So people who work on this issue, work on bacterial immunity, and when you talk about CRISPR, they tend to float towards RNAi, or RNA interference. And what is the difference between RNAi and the CRISPR system? Well, there's a fundamental difference, both in the mode of action and the target and the subsequent result. RNAi combines with the target sequence homologously and degrades the foreign DNA. However, CRISPR combines with the target sequence and cuts DNA at specific sites, very specific sites. So where RNAi degrades, CRISPR modifies. CRISPR is versatile, so it could be used to silence genes by cutting or introducing double-strand cuts and then letting the ends join non-homologously, which will effectively introduce stop codons and thus and disrupt the gene and thus render it silenced. However, also genes can be inserted using CRISPR, and this is the real advantage of the system, is that genes could be inserted. Certain genes can be inserted directly through the CRISPR-Cas system. And it's been used very widely since it's been first published as a tool in 2012, 2013. So there's an example of some knockout mice using CRISPR to knock out hair. And as a result, there has been lots of negative media about the CRISPR technology. This led even to the developer of the CRISPR-Cas9 system, Professor Doudna, to call for an international halt on research in CRISPR, because it was very evident that people were beginning to abuse it. So this is the second time in history where scientists actually call for a halt of research in a certain area. It was first called for when the ability to clone sequences was there in the early 70s. There was a consortium where they called for halting the research and reviewing the ethics. Such a call is found today, where scientists need to gather, sit down, and set the rules of the game. And it is very vital for this science to go forward using ethical means. So it's been described as scary, it's been described as inhumane, and it's been described as versatile. So lots of media attention. So to explain a little bit more in detail, the Cas protein is like Pac-Man. You know, the game Pac-Man will just eat and munch everything, unspecifically. But coupled with a guide RNA or targeting mechanism, you have a CRISPR guided missile. So it will actually cut specifically in the area of interest. So within this system, there is an identifying RNA that will homologously bind DNA at a specific site. And once the site is identified and using its helicase capacity, it will actually unwind the DNA, and using its endonuclease capacity, it will cut the DNA in double strands. And we know that in cells, when DNA is cut, panic sets in. And the immediate effect is try to find a homologous strand and try to compensate with that and wrap it up. So now I'm going to talk about the CRISPR-Cas9 system, which is the groundbreaking system. And it looks like something like this in 3D. So you have this Cas protein, and you have your guider or identifier RNA, and here is the target DNA engulfed. So it's an RNA-DNA, it's an RNA-DNA matching system, not a DNA-DNA matching system or an RNA-RNA matching system. It's an RNA-DNA matching system. And once it was discovered and developed, it immediately made the cover of Nature. This is a 2013, 14, so relatively very new. And the CRISPR-Cas9 system, which is the actual, the programmable system, was developed by these two people, Emmanuelle Charpentier and Jennifer Doudna and her team. Jennifer is from Berkeley, and Emmanuelle is in Zurich. So they looked at the naturally occurring, the wild type CRISPR-Cas9 system and found that it has the helicase, the cRNA, which is the CRISPR RNA, and the tracer, which is transactivating CRISPR RNA. And so it basically comprises of three components: the protein, the endonuclease and helicase, and then you have two RNA molecules to identify and bind the DNA. So they thought that this is a very nice system, but first of all, can we make it simpler? So they made it simpler. So they made a loop between this cRNA and tracer RNA and made a CR or CRISPR RNA, tracer RNA chimera, which made it much more stable and easier and simpler. And they thought, could we actually change this identifier sequence? Could we custom make it? Could we edit it? Could we modify it, program it? And can it cut where we want? And that's what they did. They made a very simple experiment. They made the constructs that actually change the sequence, the identifier sequence, to a required sequence. And they took a plasmid and exposed it to 4 different CRISPR-Cas9 molecules, and they wanted to see whether it cuts or not. And very surprisingly, it cut specifically at the sites that they required. A very simple, very elegant experiment, but this might as well be the first step for them towards, or they're actually being fast-tracked towards the Nobel Prize. So simple experiments can make a difference. So there we are. Now we have I think, the full set of tools in molecular biology. We used to have the tool to sequence and know the sequence. And we used to have the tool, and we had the tool to cut the enzyme, or cut the DNA using enzymes. And we discovered recently the ability to rapidly amplify DNA using PCR. But what was missing was the ability to change it, and change it reliably, efficiently, and accurately. And this is where genome editing via CRISPR comes into place. Now you can ask very simply, what's the difference between CRISPR and restriction enzymes? Do you know the difference between them from what I just explained? The difference is restriction enzymes cut at predetermined specific sites, determined by the bacteria that they generated from. Whereas CRISPR is programmable. You can actually introduce the sequence that you want to cut. And once it's cut, you can actually modify it. So it completes the toolbox in molecular biology. Now you have the ability to sequence DNA, to amplify it, to cut it at specific sites and recombine them together. And now you have a very robust tool to edit them. So this is just to show that the first publication regarding CRISPR-Cas9 came out in 2012. Nothing before that using the system, and this was followed by an explosion of research articles regarding CRISPR-Cas9. And this is up to date. I mean, I'm sure that in 2017 now, the number is astronomical. So once you have your construct, you have your ability to change and edit this CRISPR-Cas9 system at hand, then comes the mode of delivery. How do you deliver it to patients? How do you deliver it to the required system? So there's two ways. There's a straightforward in vivo approach or an ex vivo approach. So you take the CRISPR in the in vivo approach and wrap it around or package it using nanoparticles. In this case, for example, you could use lipid or carbon nanoparticles and engulf your CRISPR molecule there. And you can inject it in the blood or directly into the organs. A more safer but a more complex approach is an ex vivo approach, where you actually isolate affected cells, in this case bone marrow cells, and treat them with CRISPR, silence, edit, modify them, take them to the lab, expand them, inject them into mice and make sure that they're safe, and then inject them into the patient. So there are different ways and methods. Also, there's the possibility of actually delivering the system using lentiviruses, or modified lentivirus, a retrovirus that will ensure a massive infection of all cells, and thus having a very reliable delivery system. I ripped these graphs. I did not make them. I ripped them off the Broad Institute website. So if you've researched or Googled CRISPR, you'll recognize these graphs. So I'm sorry about that. But to date, there have been successful pilot projects that show that CRISPR is very effective in combating virus infections, particularly stubborn ones like HIV. There's a paper that was published recently of a primitive form of delivery system that eradicated around about 70% of a murine form of HIV in infected mice. So we're talking about a very primitive form, primitive system that has astronomical results. Give this 3-4 years, I think this will make a very big impact. Even in single Mendelian gene disorders, cystic fibrosis, sickle cell anemia, to name a few, have been targets using CRISPR and have been somewhat successful in modifying genes and having stable constructs to do so. But I think the most exciting and the most promising prospect of using CRISPR is in cancer. You having the ability to selectively select genes and in a multiple method. So you can deliver multiple CRISPRs at one time to cells, and having the ability to science particular genes or particular sets of genes has a great potential in cancer, because cancer is a multiple gene disorder. There are many genes that are at play. There are many genes that lead to cancer. So you can target tumor suppressor genes and oncogenes and DNA modifying genes in one go. So I'm very excited with the prospect of combating cancer using the CRISPR system. So the application of CRISPR has been growing, from human gene therapy to viral gene disruption, pathogen disruption, programmable RNA targeting, agriculture, crops management, and the list is growing. But the question is, relating to our profession, is how about diagnostics? Could CRISPR be an actual viable tool in diagnostics? Because till date, CRISPR is a very cheap system. It's a very cheap, affordable, easy. If you have basic information on molecular biology, you can do the experiment very easily. So can that be used, can that concept of easy, cheap be used in diagnostics? And up to a month ago, not really. But I was excited to introduce this talk under this title, basically because of this publication. This is a publication that came out in Science this April, and it actually shows that yes, CRISPR could be the tool for point-of-care diagnostics for several reasons. Its accuracy, its relative easiness to use, and its ability to be localized, the system to be localized and spotted on paper and delivered to remote areas, and having this tool in particularly infectious diseases conditions is very vital. So Gothenburg and Abu Daya et al. Published this paper on the use of nucleic acid detection by a different form of CRISPR, the CRISPR Cas13a, which is different from Cas9. And this team, they're from the Broad Institute of MIT and Harvard, and they used CRISPR to target RNA rather than DNA for rapid, inexpensive, and sensitive diagnostics. An RNA targeting CRISPR system rather than DNA targeting system, you can actually do RT-PCR, you can do retrovirus detection, so having an RNA CRISPR is much more versatile for a diagnostic tool than a DNA CRISPR. It is able to indicate the presence of as little as a single molecule of defected RNA or DNA. The system that we have, they call it Sherlock, and if you know the famous detective Sherlock Holmes, it's ironic, but yes, specific high sensitive enzymatic reporter unlocking system. So what they did is that they used the CRISPR Cas13a, which is different than Cas9. Cas9 is very specific. However, Cas13a cuts